As you can probably see or heard already, I won't be talking specifically about autism, but I will be talking about some of the cognitive assessment tools that we use to look at other developmental disorders that are certainly applicable to autism. So you can see that I have this long, sort of complicated, boring title, and I always have these, so I wanted to do something a little bit different. So I'm gonna replace my title real quick for everybody in something a little sexier. So when is moderation not moderate at all? And so when we're talking about moderate exposures or moderation, what we're really talking about is what the impact of prenatal alcohol exposure is long term. So we had some discussion in previous sessions about the importance of understanding what fetal alcohol exposure does. And I think it's fantastic that recently it's become fairly well known and well accepted even in the general population that high levels of prenatal alcohol exposure during pregnancy can have severe negative consequences on the physical and cognitive development of the offspring. The most severe, of course, being prenatal alcohol syndrome with its typical changes in morphology. So some clinical studies have shown that low to moderate alcohol exposure can lead to behavioral and emotional issues and impairments in executive control in adolescents, but there's other studies that have failed to find an association between low to moderate exposures and these types of behaviors. Recently, preclinical studies have suggested that even low to moderate prenatal alcohol exposures can impair certain types of executive control, in particular spatial behavioral flexibility. So despite the evidence that high levels of prenatal alcohol exposure can have major impacts, and recent evidence that even low levels perhaps can have an impact, the rates of drinking during pregnancy are still remarkably high. So as recently as 5 years ago, a report showed that 5 to 10% of women who were pregnant continued to binge drink during their pregnancy. So we'll always have a small group of people who are going to drink no matter what. However, I think the more shocking statistic is that as many as 1/3 of all women reported drinking at some time during their pregnancy. So I think it's very essential that we find out whether or not moderate exposure during pregnancy is going to have major impacts, because there's still a big misunderstanding in the general public, probably due to conflicting messages from media as well as primary care providers about what is safe to consume during pregnancy. So given reports that adolescents with fetal alcohol spectrum disorders, even not showing full-blown FAS, commonly show maladaptive perseveration, or an impairment in behavioral flexibility when they're adolescents. And given the fact that the circuits that underlie these types of behaviors are well conserved across species, we wanted to examine whether or not moderate exposure would impair executive control in a mouse model. And so the model that we're using, that was developed by Dr. Kevin Caldwell and Dr. Andrea Allen at UNM, models one of the more common exposure patterns in clinical population, and that's a first and second trimester and discontinuation in third trimester model. So how do we do this? Well, we use a limited access drinking in the dark model, where the animal is slowly ramped up from 4 days of 5% alcohol solution to 10% alcohol solution. After a 1-week acclimation period, they're given 5-day breeding rounds, and then they'll continue alcohol drinking during gestation and then weaned off immediately when the pups are born. So what do these breeding rounds look like, and what does a drinking session look like? Well, 2 hours into the dark cycle, they're given a 4-hour voluntary drinking exposure. And then during the breeding rounds, breeding occurs when applicable, and then the alcohol is removed and the dark cycle ends. So we use this exposure model, which reliably gets animals to drink to a low to moderate level. And then after weaning, we allow the animals to age to approximately 6 to 7 weeks, with no other alcohol ever given to the animals. So these animals are abstained since birth. And then we begin pre-training for some of our touchscreen tasks at approximately 6 to 7 weeks. We do a functional observation battery to make sure they don't have any gross morphological, neurological, or sensory deficits. Then they're given a discrimination problem immediately followed by reversal. So it's previously been shown that this type of this type of exposure doesn't cause any gross morphological or physical changes in the animals, which is nice because we certainly don't want to have motor impairments that are going to impair their ability and our ability to assess the cognitive function. So you can see in the data that I'm going to show you today that there isn't any changes in our full functional observation battery. I'm just showing you an abbreviated version here. In particular, there's no changes in the body weight that might affect their motivation to work for a food reward. Also, a question that I'm often asked is, well, how does the drinking affect the maternal behaviors in the dams? And I've previously shown that the dam's maternal behavior isn't altered, at least as measured by archback nursing time and pup retrieval time. So this type of model has previously been shown to impair hippocampal learning tasks, impair hippocampal neurogenesis, as well as plasticity in the hippocampus. So of course, being an executive control guy, I wanted to know, what is it going to do to behavioral flexibility? So I apologize to people who do reversal learning tasks, but I want to explain this very clearly. Because some of my clinical colleagues just do not understand when I explain what we're doing here. So it doesn't matter if you're using a touchscreen, if you're using operant levers, if you're using a maze, all discrimination reversal tasks are really alike. Is that you're going to train the animal in one type of response, and then you're going to have them shift that response when you change the reinforcement contingencies. So our responses are touches to 1 or 2 visual stimuli shown here on a touchscreen, where one always predicts reward and the other never does. Spatial orientation in this task makes no difference, whether or not your correct stimuli is on the left or right. So we've done lots of studies in normal intact animals and found that they all go through this very stereotypical process where on the first 1 or 2 sessions, they're going to randomly choose one or the other and perform right around chance. Although some animals will prefer one or the other, if you average it out, it's about chance performance. If you allow the animals to then perform the problem for about 5 to 7 sessions, they'll quickly go to a high level of criterion. The criterion we use is very high. It's 85% correct over 2 consecutive sessions. So we can look at how the animal translates from early choice to well-learned or almost automatized types of choice. So what does this look like in our male and female PAE offspring and their saccharin control littermates? Well, if we look at the total number of errors that it takes them to learn this to criterion, you can see that we have a significant main effect of sex in that all the females take longer to learn the tasks than the males. However, there's no effect of prenatal alcohol exposure in the red bars versus the white bars. The animals are able to learn the association just as well. We also use a procedure in order to overcome any stimulus bias as well as to measure perseverative behavior later in reversal, where we give them correction trials. So if an animal makes a mistake and goes to, for, let's say, this little marble array here instead of the fan, they're going to give a 10-second house light on timeout. These animals are tested during their dark cycle. And then on the next initiation, they're going to give the same stimuli and the same orientation, and that will continue as correction trials until the animal shifts and makes the correct response. So if we look at correction trials, we see that moderate prenatal exposure doesn't affect the number of correction trials it takes them to learn this initial association. Although, again, we see this sex difference on this task. Just to make sure there's no significant differences in their motivation to either respond to stimuli or retrieve their reward, we also look at the response times for both of those. And as you can see, there's no significant differences. So far we have our moderate prenatal alcohol exposure not affecting their ability to simply learn a visual response association between 2 stimuli. So what happens then with the good stuff when we reverse them and look at behavioral flexibility? So oftentimes we'll present the total number of errors or correction errors or whatever the variable is to simply get from the first session reversal back to criterion. But we actually have shown that there's 2 very distinct types of stages here. You have the stage that's just like discrimination, where you're going from chance to well-learned here in our learning stage, but you have this early phase, which we call the perseverative reversal phase, when what the animal is doing is really just going to what was previously correct, even though it's no longer rewarded. So we can analyze the data looking at how many errors and correction errors they make in this early perseverative phase when they're really having to realize that there's been a change in the reward contingencies. They have to inhibit the response and try the other stimuli. And then the later stage when they're simply learning the new association. So if we look at total number of errors it takes them, we see that in that perseverative phase, there's no difference in the MPAEs, and in the learning phase, there's no difference in the MPAEs. But what about those correction trials where they're doing bouts of incorrect responses in a row after an initial error? What we can see here is there's a significant and specific impairment in the animal's ability to get out of that early perseverative phase. In both the males and the females, moderate prenatal alcohol exposure impairs their ability, as measured by correction errors, to flexibly shift from the previously learned response to this new response. Once the animals get through that perseverative phase and simply have to learn the new association, there's no significant differences between the groups. Nicely, we don't see any differences on either stage or in either sex or treatment on the reaction time to the stimuli or to retrieve their reward, indicating that they're not simply less likely to touch the screen or retrieve. So what we seem to have is a robust impairment in behavioral flexibility after even a moderate dose of alcohol. So where is this actually occurring? If we look at correction errors by reversal session, we can see that it's really occurring as the animals are transitioning from that early perseverative phase into the chance phase and then new learning. You can see that everybody perseverates a ton when they first start because we train them to such a high level of criterion on the discrimination. The first session, they're not making many correct responses at all. They're doing upwards of 80 to 100 correction errors. So you see that the groups start to separate out here in about the fourth session of reversal as the saccharin controls nicely decrease their correction errors while the MPAEs remain elevated for longer. So this is going to be important in a little bit. So we know that this type of behavior, both the initial learning and the flexible reversal portion, are mediated in the mouse on the visual task by circuits connecting the dorsal striatum and the orbital frontal cortex. Studies using immediate early gene expression, targeted lesion of these areas, as well as genetic and pharmacological knockdown of NMDA receptor subunits have shown that these areas are critical for initial learning and the flexible component. So what we wanted to know then was, could we see dysregulations in these regions in the MPAE mice as they went through the task? So in order to do this, we used in vivo electrophysiological recording as the animal performed the touchscreen task. So we did this both in the orbital frontal cortex and the dorsal striatum in a modified touchscreen chamber that allows the animals to move freely while tethered into the system. So I just wanted to break down how we do this analysis because it can get really complicated, and if I just show you a heat map, everyone's going to tune out. So how does this actually work? So everything that goes into getting a correct response or even making an error is actually a series of behaviors. The mouse must first initiate, and we have them initiate by a bar press. Then they have to attend to the stimuli before making their choice. They make their choice, and then if that choice is correct, they retrieve their reward. If the choice is incorrect, they typically go back to the food hopper and get upset that there's no reward, and we can look at that as well. So if we take one of these events, we can look at the activity that occurs right before the event, so 1 second prior, and the activity that occurs after the event. And we found that 3 seconds is a nice time window to look at spike firing activity and not have it contaminated by whatever the next event that's coming down the pipe. So any one of these things can be an event, and the one that I'm going to talk to you about today is correct and incorrect choices. So we do the recordings in the animals, and we isolate putative neurons that are firing in the OFC, or the dorsal striatum. And we're interested in how those spike firing patterns change depending on the event. In this case, a correct choice. So if we look at those correct choice events, we'll see that some neurons in the baseline period are firing, of course. And then in the post-event portion, they're firing, of course, but they're really not changing the rate of firing. And we label those neurons non-phasic. They're active, but they don't seem to be responsive in any way that we can detect to the event of interest. Of course, there's another subset that's going to decrease their firing, and we call those neurons phasic decreasing, because if we say inhibited, we get in trouble with some of our colleagues. And we have another subset that's going to increase their firing to whatever that event of interest is. So we'll call those our phasic increasing neurons. So in addition to all 4 of those behaviors, we also have those 5 big stages of discrimination and reversal that I already laid out for you. Early and well-learned discrimination, early perseverative chance, and well-learned reversal. So now we can start building a map of what these phasic or non-phasic increasing, decreasing neurons actually look like in terms of a population of neurons. So we've got our different types, and we're going to code them. So neurons that really don't seem to alter their firing up or down are non-phasics. We'll just have them a sort of boring yellow color. The neurons that decrease their firing will code blue, and the neurons that increase will code red. In this way, we can build up a heat map of all these different behaviors as the animals go through the different phases of the task. So in early choice behavior, you can see there's really not a lot of activity during initiation while the animals are attending, and we start to see this dominance of our phasic decreasing neurons after a correct choice is made. Well, then we see a big excitatory burst as the animal gets their reward. This pattern continues and even increases as the animal goes to criterion, and these are an optimally functioning C57 animals with no alcohol exposure. So what happens then here in the OFC when the animal is required to reverse? We see a complete absence of this inhibitory phasic decreasing drive here. And keep this in mind in a little bit, I'll show you why this happens. It isn't that all the neurons are suddenly going non-phasic, but rather the average of decreasing neurons is being wiped out as phasic increasing neurons come online. As the animals get out of this perseverative phase, we see that the pattern starts to regenerate, and we see this inhibitory action followed by excitation as the OFC stops its critical function in early reversal and other systems come to dominate. So that's all well and good, but what's going on with the alcohol guys? So it takes a ton of neurons in order to build up enough data to actually see patterns like this emerge. And we just have preliminary studies in our MPAE guys. So what we're going to do is rather than look at heat maps, I'm going to show you what some of the actual neuron spike firing looks like, as well as some of the baseline. So we have these 5 different stages, and I was talking about them a minute ago, but as you might remember, I hope, from the MPAE data, the big difference is not in early choice or even in later choice, but rather in the difference between criterion performance, early reversal, and then back to chance. So we're going to call that well-learned, perseverative, and relearning, and we're going to look at what the neuronal patterns look like in these stages. So what does the phasic baseline firing in the OFC look like in these animals? What you can see is that during well-learned, there's no significant difference between the groups at all. The baseline firing before the choice event is no different. When you get into early reversal, you see an increase in the saccharin control animals in the baseline phasic firing that's completely absent in the MPAE animals. By the time they get to the relearning phase, this pattern is actually reversed. We sort of see a delayed increase in the phasic baseline firing in these MPAE guys. So if this is baseline then, what is going on in terms of the neurons that are phasic post-event? Well, we can't look at a heat map because we simply don't have enough neurons yet, but we can look at the proportions of neurons that are increasing or decreasing their firing. So you can see in well-learned that the majority of saccharin animals are inhibitory, the phasic neurons that are seen there, while in the MPAE animals, we have a balance towards excitation even in the well-learned phase. We see that the OFC neurons shift to much more phasic increasing that occurs in the PAE animals as well, but to a much less extent. That goes away by the time they get into the relearning phase, but of course we have this delayed increase in our MPAE animals. So we can also look at what the pattern looks like of those neurons that are increasing and decreasing. We have a population in our saccharin animals that significantly decrease, another population that significantly increases, and the MPA looks pretty much the same. So although the proportions of neurons that are increasing or decreasing differ, there's no difference in the pattern. So what's going on then in the dorsal striatum if the OFC seems to be dysregulated in these patterns? Well, you can see there's no difference in the baseline phasic firing here, and there's a significant decrease in the dorsal striatum firing. In the saccharin controls, it's completely absent, the exact opposite pattern than what we see in the OFC. And then by the time they get into the relearning, we have this giant jump, which may be due to the small n in these studies. We see that throughout all the stages, sorry, I'm running out of time, that the dorsal striatum just seems to be ramped up in every possible direction. So as opposed to the decreases in the OFC firing that we see in the MPAE animals, we see just an incredible burst of activity in the dorsal striatum in these animals, indicating that as in our chronic binge model, they may be more primed to be habitual, although we don't have data showing that at this point. Probably most interesting, because this is still a fairly low N, and I was shocked that we were able to see any patterns, we have in the saccharin animals a proportion of dorsal striatal neurons that significantly decrease. We have a proportion that significantly increases. We have a proportion in the MPAE that decrease, and maybe a slight increase in the decrease, if I'm allowed to say that. But when you look at the neurons that are increasing their firing, you see a significant and robust ramp up in the MPAE dorsal striatal neurons. So with this in mind, the studies that we're undertaking right now are to see is whether or not we can alter the OFC firing to increase it to normal levels in order to try to rescue the maladaptive perseveration that we see in these animals. So the phase that seems to be most important here is this choice phase, and the timing that seems to be most important is sessions 4 through 6 of the discrimination reversal problem. So in order to do this, we're now adding an optical fiber to our in vivo arrays, and that's what it looks like. You can see that we have a bundle here with an optical fiber targeting the center of the bundle. And we're also using a lentiviral vector to express channel rhodopsin in the orbital frontal cortex. And we have some proof of principle data showing that we can isolate neurons here, and we can significantly increase the firing of the OFC neurons when we give them the light stimulation online while they're active in the touchscreen boxes. So the current directions that we're having, as you might have surmised already, is if it's so important to look at these first 6 reversal sessions, why are you recording throughout all these other different stages? So what we're doing now is shifting from a pattern where we will record in either the dorsal striatum or the OFC during these key stages of discrimination reversal, which is a fantastic strategy if you want to look at optimal functioning, to recording in both the DS and the OFC of the same animal so that we can look at how these two regions are interacting as the animal does these behaviors. And we're front-loading all of our recording into these critical phases that are affected by moderate prenatal alcohol exposure. So I just want to summarize that we see that MPA significantly impairs behavioral flexibility by selectively increasing maladaptive perseveration. And our preliminary data suggests that increases in OFC firing that are required for reward updating are simply not there in these MPAE animals. We think that this may be leading to an increased striatal firing that's driving this perseverative behavior, and we're currently experimenting on whether or not we can selectively increase the OFC activity in order to rescue this maladaptive perseveration. So with that, I want to thank Kristen Marquardt, the graduate student in the lab, and my technician, Megan Josie, who've been really, really wonderful in the in vivo recording experiments and the optogenetic experiments. My colleagues at UNM and collaborators, and thank you to IBNS for the opportunity to be here.